So we are now switching over to focus on viruses that infect animal cells. You can apply this to human cells, but this is just more generically speaking to animal viruses. And so the same 5 steps that we learned are going to be in play. And so the first is attachment. These are all in the previous slides, but I'm just going to put them here so we have them. And then entry, then synthesis of those viral parts, and then assembly of those parts, and then somehow the virus is going to be released. So we're not really changing anything from what we saw previously, but we just want to understand and have it geared towards viruses that infect animal cells. And so the first thing we're going to look at is these first 2 steps kind of collectively. So the same information still is true, is that there are molecules on the surface of any, so again, the term that we use frequently is virion, that infectious viral particle. There are molecules on the surface that has a unique shape to them. So they're often called viral spikes, and you can think of them as having unique shapes, much like keys. And that allows for the binding of a virion to a host cell. So this first picture here, this is a host cell. It's a zoomed in view so you can't see it. This would be the outer edge. This would be the cell membrane, so you can see the relative size difference. This would be the example that's being shown here. This is a naked virus. So there is no envelope that's being shown, but this is a capsid. More specifically, it's a polyhedral capsid inside of which there's nucleic acid. But these are still proteins on the surface. So there are viral proteins that make up the capsid, and they have a unique shape that can allow them to interact with molecules on the surface of the cell. And so that's what we're seeing going on for attachment. So we saw that for the bacteriophage as well. This particular route of entry, it's called direct penetration. And so molecules on the surface of this capsid is going to really basically bring the virus in really close and interact with this cell membrane, basically create a small pore that can allow the viral genome, that nucleic acid, to penetrate that cell membrane directly, and now you have nucleic acid inside the cell. So virus, nucleic acid inside cell is what we can say. And so that's what direct penetration can allow a virus to do. So some viruses utilize this mechanism of entry. I didn't say this at the beginning, but there are 3 different ways that viruses can enter a human cell or animal cells. So direct penetration is one of those. More common is membrane fusion, and membrane fusion can only work only for enveloped viruses. So if a virus is enveloped, it could use this mechanism of entry. And so this is an envelope virus. You can kind of see this sphere that encircles it. If you look closely, there is a capsid inside. You can see it better now because they've cut it away. There's a capsid inside that. And then inside that capsid is viral nucleic acid. But attachment starts the same way. Viral glycoproteins, those are called viral spikes also. I don't know what happened to my writing there. Spikes. That allows the envelope virus to bind to molecules on the surface of the host cell. So that's what we see there. And you can see how closely they're interacting there. So this would be the viral protein, and it fits perfectly in that cell surface receptor there. So that's attachment. And then during membrane fusion, what's going to happen is you can actually see the 2 membranes. So that's the cell membrane. I'm just going to abbreviate cell membrane. And then this is the envelope. And so those two phospholipid bilayers are right on top of each other. And there are molecules in this envelope that are going to trigger the fusion or the blending of those two layers together. And so that's what happens between these two. And so when that occurs, what you get is the capsid is now on the inside of the cell, and inside the capsid is the viral nucleic acid. And so as we mentioned previously, what happens at the end of entry and the way that the virus's intracellular structure has to be is only nucleic acid, so we're not quite done yet with the entry. And so there's one more step that has to happen. This capsid, also called a protein coat, has to be removed. And so that process is called viral uncoating. And so then what you get is there is only the viral genome or the viral nucleic acid. Okay, so that's the entry step. So it's called membrane fusion, and that can only be used for envelope viruses. And then the last, so this is the final of three entry mechanisms that can be used by human viruses. It's called endocytosis. This can be used for either naked viruses. So if it's a naked virus, it can use this method, or envelope viruses. So really any virus could potentially use this enveloped. So the example that they're showing here is some envelope virus, because again here we can see that's the envelope, and then this is the capsid again. They like to show polyhedral for whatever reason, but it could just as easily be helical. And then inside the capsid is viral nucleic acid again. So attachment starts off by having those keys. So viral molecules on the surface of the envelope interact with molecules on the surface of the cell. So again, think of a key and a lock. So that's the first step. But that's also going to trigger the second mechanism. So this term might be familiar to you because it's a normal process of how things enter a cell. So backing up a little bit and just reminding you of the function of the cell membrane is it's a barrier. It serves as a protective layer that keeps things out of the cell and keeps things in the cell, but it also has mechanisms that allow things to move in and things to move out. And so one of those mechanisms of importing things into the cell is called endocytosis. So it's a normal process. Viruses didn't invent this. They just have learned a way to hijack it for their own purposes because they have to enter a host cell. So why not utilize a mechanism that's already in place? And so that's indeed what they do. So when the virus comes in close contact, in close proximity to the cell, then there can be additional molecules on the surface that will trigger this process of endocytosis. And what happens is you can see the cell membrane start to almost wrap around and completely surround. So I'm highlighting the cell membrane right now. And this is going to continue until all of these basically run into each other and completely surround it. And so what you're going to get is basically a piece of the cell membrane pinching off. So this outer layer is derived from the cell membrane, and then this inner one was that viral envelope. So it's kind of double wrapped in membrane right now. If you notice, here was just a single phospholipid bilayer, but now another whole one wrapped around it, and that's what endocytosis is. But the continuing steps of endocytosis would be to remove that lipid outer layer, and so now you have viral capsid, and inside the capsid is the virus, nucleic acid. And so at the end of entry, we still only want viral nucleic acid, so that uncoating step has to happen again. So regardless of which of these three mechanisms is used, what you get at the end is there is viral nucleic acid that is now inside the cell, whereas prior to this it wasn't present. And so that's what happened during those first two steps of attachment and then entry. I don't know if I've said this specifically, so I just want to make sure we're clear on it. For any given virus, they only use one mechanism of entry, so they don't change it depending on the situation. So there's not a virus that sometimes uses membrane fusion, other times uses endocytosis. They have evolved one mechanism of entry, and that's what they use each and every time. So the three that we've listed are just the three that we know that exist. So any virus will be put into one of these three entry mechanisms. That's how it works. So after entry again, regardless of which of the three mechanisms we're talking about, what you have is the virus's genome, its nucleic acid, which is again either DNA or RNA, in that fluid-filled stuff of the host cell that we call a cytoplasm, where this says cytosol. So those are the first two steps. I'm not going to rewrite them all, but step 3, now that there is viral nucleic acid present, or a viral instruction manual present, that next step of synthesis can take place, and so that's what we're going to start to talk about. So we're not going to go into detail with this, and so we're going to talk kind of just general things. So synthesis is again going to require understanding what is the virus trying to make. And so the end goal is to make those things called virions, those infectious viral particles. And so if you understand their structure, we know that they require proteins and they require nucleic acid, because a virion is going to be a protein coat or capsid that surrounds nucleic acid. And so synthesis is very much going to be driven by the making of viral proteins. And so all viruses have to have a solution to what is going to be used as that messenger RNA, because it's the messenger RNA that directs protein synthesis. And then what is going to be used as basically, we call it the template. So we see that term often when nucleic acids are used to make more nucleic acids. So there has to be some template molecule that's used to be copied over and over again to make that viral genome. So that's where it starts to get a little bit more tricky, and it differs depending upon what viruses we're interested in talking about. So we're going to keep it really generic and just realize that that is the situation that all viruses have to solve to be successful. They have to make proteins because they found some way to make messenger RNA, and then they have to make nucleic acid. The answer to how these questions are solved varies tremendously across different groups of viruses. So that goes beyond the scope of this class. But what we can also say is, depending upon what type of virus, we can talk some more generalities, because viruses use host machinery. So if there's a DNA virus, where all the machinery exists to kind of work on DNA is in the nucleus of our cells. And so DNA viruses, for them to be successful, frequently have to enter the nucleus so they can gain access and utilize all that machinery to copy viral DNA into more viral DNA, for example. Whereas RNA is used in the cytoplasm of the cell. And so often, if it's an RNA virus, they don't have to enter the nucleus because the machinery that they kind of plug into exists in the cytoplasm. As I mentioned, there are a variety of different ways to solve these two major hurdles that any virus has to do. How is mRNA going to be synthesized to make proteins? And what's the template that's used to make more viral nucleic acid? You don't have to memorize this. Really, what I just want you to realize is that there are different solutions for different groups of viruses. So if you look over here on the left, these are different ways that the viral genome can be organized. So remember, DS, double-stranded DNA, SS, single-stranded DNA. So single-stranded RNA, and then double-stranded RNA. So depending upon how the virus's genome is organized is going to then lead to different solutions. And what those solutions are as I mentioned before, are beyond the scope of the class. But viruses have found a way to do it. So kind of without going into great detail on how that's achieved, viruses do make the viral parts. And so then we get to the last two steps. So step 4 would be assembling of the viral particles, and then how are they released. So assembly, as you might guess, depending upon where the parts are. So DNA are often assembled in that nucleus, because that's where DNA is being copied. And then RNA viruses are often assembled in the cytoplasm, because that's where most of the RNA machinery is. And depending upon the virus, there's different routes of release. So it's possible, just like in the lytic cycle, that viruses can just kill the cell and lyse it. So that works. This term might be familiar because it looks almost exactly like endocytosis, but notice it starts with exo. So that prefix just is going to relate to exiting as opposed to endo, which means going in. So exocytosis is kind of the reverse of endo, and so this is a process that cells use to have things leave the cell. So viruses have found a way to hijack that. There is a process, I'm looking to see if I have it written on here, that we're going to see. It's called viral budding, that we're going to look at. And so that is a process, and it's an explanation of how some viruses can leave the cell. And it can also, budding can also explain this statement where envelope viruses that leave cells by budding, what you're going to see is it doesn't kill the host cell. And so that host cell can maintain a viral infection for an extended period of time, because new viruses can be shed from that cell persistently over a consistent, length of time. Whereas if the virus causes lysis, then that cell can no longer make new viruses. So it's kind of one massive release event upon the lysis of the cell. But then that cell no longer is going to synthesize and assemble any more viruses after that point. So when we look at budding, which is what this slide is, it's going to explain kind of two things. So budding is going to answer the question of release. So this is explaining how a newly assembled virus can leave the cell, that 5th step. But it also explains how an envelope is obtained. So as we mentioned, an envelope virus is going to derive that phospholipid envelope layer from the host cell. Okay, so what we have here, so this is inside the cell. That's in, this is out. And so we're again looking at that cell membrane. Here is an assembled. So we've gone through that assembly process. It's got a polyhedral capsid on it. You can see the nucleic acid on the inside. And it's going to interact with the cell membrane from the inside. And as it does so, you can see it looks like that cell membrane is being draped over or starting to wrap around. So this is the process of budding. And this is going to continue where you can see that cell membrane is starting to pinch together until it totally pinches off. And so now not only do we have this virion on the outside of the cell, but it now has the viral envelope encircling it. So now we have an enveloped virion that can go find another host cell to infect, and the process would then continue with the first step of attachment, and then entry, and then synthesis, and then assembly, and so on. So that's what we have. One thing to note, this is really to put a pin in this for really later. Notice what we have here. Because the goal is to have these unique viral proteins on the surface of the envelope, we call them viral spikes or viral glycoproteins. So for them to get onto the envelope, they have to be on the cell membrane as that cell membrane is being acquired as the envelope. And so these proteins on the surface of this cell are unique viral proteins. So another way of saying that is these proteins will not be present on every cell in our body, but just this virally infected cell or any other cell that is infected by the same virus. The reason I'm bringing this to your attention now is because this is a really important part of how our body fights viral infections. So if you think about how our immune system targets and destroys things, they need to be able to recognize differences between our own healthy tissue and cells as opposed to what is an invader like a bacteria or a virus. One of the problems with viruses, as hopefully you kind of already figured out, is they're frequently inside our own cells. And so how can our immune system find and identify a virally infected cell compared to a non-infected cell or an uninfected cell? And part of the answer is having these viral proteins on the surface, which is a necessary part of the viral life cycle. So imagine if these weren't present, then that means you would have a virion with an envelope, but it has no keys on the surface. That's not going to be a very successful virion. So these are really critical to how this virus is going to be operating. But that also now allows for our immune system to potentially identify this as being foreign and maybe target this cell for destruction during certain immune responses. So that's just planting the seed for a later discussion when we get to it towards the end of the semester. One other thing we can talk about is this term that's in the title of the slide. It's called latency. And this is really similar to that lysogenic cycle that we saw earlier. So it's been some time, but if you go back and look at that slide, a virus entered the cell. So step one, it attached. Step two, it entered. But then it didn't synthesize viral parts right away. It kind of pressed pause in the life cycle and just remained dormant, so to speak, inside of the host cell. And that's what viral latency can be. So prolonged, it can be for many, many years. And so there's no signs of viral activity. There may be no viruses being shed from the patient, no signs of disease, no symptoms of disease. And so latency is when that virus is remaining basically dormant inside that host cell. There's different mechanisms of how this can be achieved. So it's possible that the DNA, sometimes it just sits in the cell by itself, so as a separate piece of nucleic acid, separate from our chromosomes. But there are instances where the virus DNA integrates or incorporates, and so we use this very similar term. We called it a prophage in the lysogenic cycle, but we call it a provirus if the virus can integrate its genetic material into our own. So this is a permanent change to our genetic material because once the DNA integrates, the viral DNA integrates, it is now part of our chromosomes. So there are certain viral diseases that can remain latent. So human papillomaviruses can remain latent for extended periods of time. So too can herpes viruses. That in part explains why if you've had chickenpox, it's possible for you to have a different disease manifestation later in life that's called shingles. It's the same virus, but it has to do with the latency and then the reactivation of it. But we'll talk about individual viruses later on. But the goal of this lecture was really to introduce you to the general information that you can use to really help understand any virus now.